Hello dear students, welcome to Rose Project. Welcome to this online video lecture conducted by Rayat Online School Education, that is the Rose Project. Myself Miss Patil SD from HM College Karad. I am conducting here this video lecture from biology. The topic is plant-water relation. In last lecture, we studied the points like plasmolysis, then mechanism of absorption like active absorption, passive absorption, then pathways of water, etc. Today's point is transport of mineral ions. First point, soil serves as a main source for minerals. Second, minerals constitute most commonly occurring solid, crystalline, inorganic materials obtained from Earth's crust. Next, minerals plays important role in the day-to-day life of a plant. Next, minerals are absorbed by plants in the ionic form mainly through roots and then transported. Next point, absorbed mineral ions are pulled in upward direction along with xylem sap because of transpiration pull. Mineral ions are needed in the areas of the plant like apical, lateral, young leaves, developing flowers, fruits, seeds, and storage organs. Go to the next slide. Plants absorb inorganic nutrients from soil, which acts as the reservoir of mineral elements. The study of absorption and metabolism of inorganic nutrient is called mineral nutrition. This is the definition of mineral nutrition. Next, the minerals that play important role in the day-to-day life are called essential elements. About 30 to 60 elements are incorporated in the plant's life. Next, some minerals like carbon, hydrogen, oxygen, phosphorus, nitrogen, sulfur, magnesium, calcium, potassium, etc. are required in large quantity are called macro elements. The elements which are required in large quantity are called macro elements, while some minerals like copper, cobalt, manganese, then boron, zinc, iron, molybdenum, chlorine, which are required in small quantity are called micro elements. Go to the next slide. The next point is transport of food. All the plant parts require a continuous supply of food for the nutrition and development. Second, in higher plants there is a great differentiation and the division of labor. Chloroplasts are confined to green cells of leaves where food is synthesized. In leaves, food is synthesized. The non-green parts like root and stem must receive the food from leaves. The parts where the food is synthesized is called source, while the part where it is utilized is called sink. Food has a travel from source to sink. In this point, in the source where the food is synthesized means prepared, which is called as source, and the site is called as source, and where food is utilized which is called as sink, and then food is traveled from source to sink. This movement of the food from one part to the other part is called translocation. Translocation of food. Go to the next slide. Path of translocation. First, food is to be translocated to longer distances in higher plants. Hence, the plants must have adequate channels for the transport of food. Second, sieve tubes and vessels are structurally ideally suited for longitudinal translocation, means vertical. Next, the ringing experiment structure and distribution of phloem, chemical analysis of phloem sap, and use of isotope 14C clearly point out that the phloem tissue is primarily responsible for flow of food in longitudinal downward direction. Go to the next slide. The horizontal translocation occurs from the phloem to pith or phloem to cortex via medullary rays in the stem. When some trees are larger, then in lateral position, how food is transported with the help of medullary rays. Medullary rays also responsible for translocation of food in large surface area of the trees. Next, food is always translocated in the form of sucrose and always along the concentration gradient from source to sink. The transport of food occur in the vertical and lateral direction. Go to the next slide. Vertical translocation. First point. In vertical transport, food is translocated in the downward direction from leaves to the stem and root. Second, it also occurs in upward direction during germination of seed, bulbils, corn, etc. In vertical translocation, here the also food is translocated downward direction means that from leaves to the stem and root. Also in upward direction towards the germination of seed. Upward translocation also occurs from leaves to growing point of stem to developing flowers and fruits situated near the ends of the branches of a stem. Next slide. Here it is the diagram of longitudinal section of sieve tube. These are the transporting tissue, xylem tracheids and xylem sieve tubes or sieve cells. See here, it is the sieve plate. Then when food material is goes to the downward direction, which is goes towards the root, and also in upward direction it goes towards the stem and leaves. Next, lateral translocation. First, it occurs in the root and stem. When food is translocated from phloem to pith, it is called radial translocation, and from phloem to cortex, it is called tangential translocation. Next, the transport of food through phloem is bidirectional. Next, the phloem sap contains mainly water and food in the form of sucrose, but sugars, amino acids, and hormones are also transported through phloem. Go to the next slide. Ernst, Ernst and Munch proposed that the photosynthetic cell synthesizes a glucose. Hence, its osmotic concentration is increases. Due to endoosmosis, water from surrounding cells and xylem is absorbed. The cell becomes turgid. When endoosmosis occurs, the cell becomes turgid. Due to increase in turgor pressure, sugar from photosynthetic cell is forced ultimately into the sieve tube of the vein. This is called as loading of vein. Root cell utilizes sugar and polymerizes excess sugar into the starch. Its osmotic concentration is lowered and exosmosis occurs. Go to the next slide. Water in the root cell is lost to surrounding cells, thereby decreasing the turgidity of cell. Then turgor pressure is lowered, means decreased. Hence, the turgor pressure gradient is developed from sieve tube and in the lip to the root cell. Due to food is translocated along the concentration gradient passively. This is called vein unloading. Next slide. Here it is the transpiration process which occur in plants. When water is absorbed by the roots, see here, roots take up a water from the soil. Then water is drawn up the stem to the leaves. Here it is the stem and then here leaves. Water is goes towards the stem and also to the leaves. Here veins carry water into the leaves. Then with the water evaporates from the leaves. This process is called as transpiration. When the loss of water in the form of water vapor, this process is called as transpiration. Go to the next slide. Transpiration. First point. Plants absorb water constantly and continuously. Only 5% of the total water absorbed by roots, that is utilized for a cell expansion and plant growth. Remaining 95% of the water becomes surplus, which is then lost into the atmosphere through its aerial parts. Hardly 1% of surplus water is lost in the form of liquid and 99% of surplus water is lost in the form of vapor. The loss of water in the form of liquids is called guttation, and it occurs through a special structures called water stomata or hydrathodes. Next slide. The loss of water in the form of vapor is called transpiration that occurs through leaves, stem, flowers and fruits. Most of the transpiration occurs through the leaves. In transpiration, loss of water in the form of vapor, this process is called as transpiration. Next, the actual water loss during transpiration occurs through and it consists of three types of transpiration, that is a cuticular transpiration, then second stomatal and last lenticular transpiration. In that first one, a cuticular transpiration. In that first point, a cuticle is a layer of waxy substance, cutin, which are present on outer surface of the epidermal cells of leaves and stem. The cutin, which is a waxy substance which are present on a epidermal side of the leaves and stem. Second, cuticular transpiration occurs by a simple diffusion and contributes 8 to 10% of the total transpiration. Means out of the total transpiration, only 8 to 10% transpiration which takes place through the cuticle. Cuticular transpiration occurs throughout the day and its rate is inversely proportional to thickness of the cuticle. Next slide. Lenticular transpiration. Lenticels are small raised structures composed of loosely arranged complementary cells. Second, each lenticel is a porous tissue consisting of cells with large intercellular spaces in the periderm of the secondarily thickened organs and the bark of woody stems and the root of dicotyledonous flowering plants. Next, the lenticels are present in a bark of old stem and pericarp of woody fruits but are absent in leaves. It contributes only about 0.1 to 1% of total transpiration. Out of the total transpiration, only 0.1 to 1% transpiration takes place through the lenticels. Rate of lenticular transpiration is very slow. It also occurs throughout the day. Next slide. Third type of transpiration is a stomata transpiration. In that, first, the stomata are minute apertures formed of two guard cells and accessory cells. 90 to 93% of the total transpiration occurs. Here, 90 to 93% means out of total transpiration, most of the transpiration takes place through the stomatal transpiration, through the stomata. Second, they are located in the epidermis of young stem and leaves. Leaves generally shows more number of stomata on the lower surface. Depending upon the distribution of stomata on leaves, it consists of three types, namely epistomatic, then second hypostomatic and third amphistomatic. Epistomatic means the stomata are present on upper epidermis. Example hydrophytic plant, example lotus. Then hypostomatic, which are present on lower epidermis in xerophytic plant, example nerium. And third, amphistomatic, which are present on both surface in mesophytic plant, example grasses. Stomata transpiration occurs only during daytime. Exceptions are desert plants. Go to the next slide. Structure of stomatal apparatus. First point, a typical stomatal apparatus consists of two guard cells, stoma, and accessory cells. Second, the stomata are minute elliptical pores bounded by two kidney or dumbbell-shaped guard cells. Guard cell is a type of epidermal tissue which may be called as modified epidermal parenchyma cell. Second, they are kidney-shaped in dicotyledonous plants and dumbbell-shaped in monocotyledonous plants. Here, the next slide is the structure of guard cells. In this diagram, we show these are the epidermal cells and this pinkish color which are the subsidiary cells. Here, it is the inner thick wall and outer thin wall. These are the pore of stoma and it is the pinkish color. In guard cells, it is the nucleus. Okay. Second, it is the another diagram means type of guard cells. First, it is the kidney-shaped guard cells which is present in monocotyledonous plants, and here it is a dumbbell-shaped guard cells which are present in monocotyledonous plants. Okay. Next slide. In Cyperus, both kidney and dumbbell-shaped guard cells are present. It is very important line according to the exam. In Cyperus, both kidney or dumbbell-shaped guard cells are present. Then next, guard cells are living nucleated cells with unevenly thick walls. Inner wall of guard cells is thick and inelastic, and its lateral wall is thin and elastic. Guard cells contain few chloroplasts which are capable of a poor photosynthesis. They have ability to change their size and form due to which stoma opens or closes. Next, stoma is an elliptical pore formed due to specific arrangement of guard cells. It is through the stoma excess water is lost in the form of vapor. Go to the next slide. The second cells are accessory cells. These are specialized epidermal cells surrounding the guard cells. Their number is variable and are reservoirs of K+ ions, means potassium ions. Second, they are also called as subsidiary cells. Subsidiary cells, accessory cells which are also called as subsidiary cells. Go to the next slide. Mechanism of opening and closing of stomata. In during daytime what happens, and here during night time what happens. See, in during daytime or also in the presence of sunlight. Here the CO2 concentration decreases, then malic acid formed in guard cells then dissociates into malate ions and H plus ions, then K plus ion exchange from subsidiary cells, then osmotic pressure of guard cell increases, then endosmosis into guard cells. Means here endosmosis process occur and then cell get turgid and then stomata are opened. Here this process is occurring during daytime. And second, during night time, CO2 concentration increases, then abscisic acid means ABA, which are released to the K plus exchange, then K plus ions transported back into the subsidiary cells. Means here subsidiary cells which are reservoir of K plus ions. Then decreases the pH of guard cells, then starch synthesized in guard cells, then decreased osmotic pressure of guard cells, then exosmosis from guard cells. Means guard cells get shrinked and then stomata are closed. These processes which are also occurring at a night time. Next slide. Advantages or significance of transpiration. In that first point, it removes excess of water. Second, it helps in the passive absorption of water and the minerals from the soil. Third, it helps in the ascent of sap. Next, as the stomata are open, gaseous exchange required for photosynthesis and respiration is facilitated. Next, it maintains turgor of the cells. Next, transpiration helps in the reducing the temperature of the leaf and in imparting cooling effect. Disadvantages. Excessive transpiration leads to wilting and injury in the plant. It may also lead to the death of the plant. Means that excessive transpiration means more transpiration which is not benefited to plant body, which cause the death of the plant. Okay, thank you.